Good morning or good afternoon, everybody. Welcome to our webinar. So we're happy to start today to talk to you about BioBRIDGe Awards. So we're with the Critical Path Institute, and we are the Translational Therapeutics Accelerator, or TRxA program. And today we would like to tell you about our biologics-focused Bridging Research and Innovation in Drug Development Grants, or BioBRIDGe Awards. First, I'd like to introduce the team to everybody that's joining us today. So let's kick things off with Mark.
Yeah, hi, my name's Mark Drew. I'm the Director of Drug Discovery and Development. My background's in pharma, mostly at Sanofi. I'm a small molecule medicinal chemist.
Wonderful. And Michelle.
Good evening, everyone. I'm Michelle Morgan. I'm Associate Director of TRxA. I oversee the operational aspect of our program. Welcome, everyone.
Thank you. And Kyla.
Hello, thank you all for joining us today. My name is Kyla Edding. I'm Project Coordinator for TRxA, and I work closely with Michelle on the operational side.
Thank you. So yeah, we have a great team. My name is Micah Averts. I'm the Executive Director of TRxA. Joined CPATH about 2 years ago when we initiated the TRxA program. Background in pharmaceutical science, pharmacokinetics, and drug delivery in particular. And then I spent a lot of my career at the University of Alabama at Birmingham, where I also led an academic drug discovery program called the Alabama Drug Discovery Alliance. But very excited to be at CPATH and with TRxA, where we'll talk about in the next hour what we do here to support you. So our mission is to be a global drug discovery and development program, and we support academic scientists to advance new cutting-edge therapeutics from the lab to the clinic. So there's a couple of words highlighted in this statement. First is global. So we actually can support projects all over the world. It doesn't matter where it is, as long as it's a good project and it fits within our parameters, we are happy to support it. We do only support academic or nonprofit scientists. We do not support startups or small companies. Just wanted to make that clear from the outside. And we'll talk a little bit more later why that is. And then we focus on supporting therapeutics development. So not, for example, vaccines or devices. Again, something that we'll talk a little bit more in the next slide. So our current focus is to support therapeutic molecules, like we just said, and they need to be in the lead optimization IND-enabling studies stage. So we do not support early stage discovery, for example, high throughput screening or basic science. We also do not fund or support anything clinical, but that in between stage. We are looking for projects with composition of matter IP either in place or obtainable. So this means that repurposing does not fall within our purview at this point in time. And then the intellectual property still needs to be associated with the institution, whether it's a university, research hospital, research institute, a nonprofit entity of some sort. Again, speaking to that, we can only support projects in a nonprofit sector. So why do we exist? So probably many people on this call are familiar with the valley of death. So resources are in the early stages in academia are fairly, I wouldn't say plentiful, but with support in the United States, at least through the NIH, there's quite a bit of support for basic research. On the industry side, once you get to the clinic, there's quite a support as well. But this in-between stage is called the valley of death. So in academia, there's a lot of basic science being done. In industry, typically they end up supporting a project through the clinical phases, but this middle piece is called a translational aspect. And this is really where there's a couple of aspects that come into play. Why is this such a hard problem to tackle? So there's a couple of points here in the box at the bottom. Regulatory aspects, and I'll speak to those a little bit more. How do you know if it's clinically relevant, what you're doing in academia? Is it going to make a difference ultimately in a patient's life? What kind of technical expertise do you need to kind of traverse this valley of death? There's some areas of expertise that we're typically not exposed to in a university setting, but industry scientists are very well aware of, but how do you bridge the communication and technology gap? Reproducibility, we'll talk about that a little bit more as well. Collaboration with team science becoming more and more hip or important, I would say, in academic science. There's more emphasis on it, but still in academia, typically it's still very individualized efforts within single labs. But for this translational science, you need, let's say, somebody who knows the biology, a clinical expert, somebody who understands manufacturing, maybe a chemist. And it's all those people that need to come together in these collaborative teams that get something through this value of death. Risk is a big issue as well. When institutions like venture capital groups or industry want to take a project out of academia, they would like to see it as de-risked as possible. And then funding is an issue as well. So I'll try to talk about a couple of these issues and how TRxA and accelerators like us can help. So the first is regulatory science aspects. So there's a nice graphic here on the right, which speaks to the FDA, although we also work with other regulatory agencies. And their mission is to protect and advance public health. And regulatory science is the science to develop tools, standards, and approaches to assess the safety, efficacy, quality, and performance of regulated products. Well, what does that mean for drug discovery and development? Well, when early phase scientists in academic institutions and nonprofits think about how to discover a new drug, there's typically a lack of awareness around these tools and concepts. For example, it would be really good exercise to put together a target product profile, or TPP. And this is a document that lays out what is the desired route of administration, the patient population, what are acceptable side effects, how often do you need to take this drug? Is it once a day dosing? Is this an injection every 3 months? And you need to have conversations around the ultimate user, which is the patient, and the care providers to understand what needs to go into this target product profile to have something that ultimately will make a difference in the lives of patients. And then there's a practical aspect of how do you even submit an IND? An IND stands for Investigational New Drug Application. Some universities have some support offices to help you with that, but not all. And it's really a black box a little bit, like what's needed to submit an IND to the FDA to get your product or your drug to a stage where the agency is comfortable letting it be administered to a human being. And what are appropriate clinical endpoints? How would you design a clinical trial? And if you're a basic biologist, you're like, well, is that really my problem? Well, having the conversation may actually inform how you are going to set up your animal experiments to test if your drug in development is going to make a difference. And there's also support programs that regulatory agencies provide that having an awareness of those will actually maybe make your life better. So there's an example from the FDA. They started the Oncology Center of Excellence, Project Catalyst. And in that particular project, they facilitate what they call accelerator innovator discussions. There are meetings between the agency and the scientists to together brainstorm, okay, what do you need to do? There are more informal meetings than typically you would have with the FDA. And there's also an oncology regulatory expertise and early guidance learning platform through this project, where investigators can go to the FDA website and do a self-paced learning about what actually do I need to do from a regulatory perspective. An example from the EMA, or European Medicine Agency, is that the fee for scientific advice, which normally you have to pay for, but it's waived if you are doing an orphan and pediatric indication. Many people on this call probably work on pediatric or rare and orphan diseases. So knowing that you actually can go to the EMA and for free get advice, that's a really useful thing to be aware of. And I want to speak briefly about reproducibility being an issue. So there's a publication that came out a few years ago. It's on the right with I'm sure a clickbaity heading of why most published research findings are false. And it's not because there's evil scientists doctoring all kinds of data in the lab. Of course, there's examples of that, but it's not the majority. It's really when there's a small sample size or when there's a small effect size that translates into the research finding being, there's a good chance it's not going to be reproducible. If there's a lot of flexibility in how you design an experiment, how do you define what the outcome needs to be, how do you analyze it? Same thing, reproducibility is an issue. And then there's cultural issues when there's great financial interest, prejudices, or if the scientific field is really hot, then there's also been a statistical correlation between reproducibility being an issue. But those first couple of points really speak to robustness of the research enterprise, which sometimes is lacking. And it was a really funny cartoon in a Nature drug discovery article a few years ago, where it's like, spot the difference. So on the left-hand side, you have an experiment being done in a lab in June with a couple of different lab people, and on the right, same experiment being done in October. The weather is different, the people are different. It looks a little bit more sloppy, to be honest. But if your experiment can be repeated by a different group at a different time, that really speaks to how robust your data is and how de-risked it would be for a potential investor that's coming down the line and wants to invest in your project. So speaking of de-risking, accelerators like TRxA can help with some of this as well. There's more accelerators, not just TRxA, exist out there. But in general, there's a couple of approaches that accelerators bring to the table to really help with this de-risking and making sure that I's are dotted, T's are crossed, so that a venture capital group or a potential pharmaceutical company or biotech has confidence that what you're seeing is real, and that they have a good chance of reproducing it then in their hands and then build on that. So approaches that accelerators bring to the table is this organizational culture of collaboration, this interdisciplinary project team that you need to put together with all those different layers of expertise, having all those eyeballs on the project to anticipate problems that may be coming down the line. For example, scale up. How are you going to make more of your drug? Actually taking that into account when you're still on the front end of your discovery phase would be very useful. And then there's going to be diligence around target selection. So many accelerators, including ours, have an application process, and there will be a review like, okay, why is your target important? Is it druggable? What is the role of this target in a disease? And has it been clearly defined? Have there been genetic levels of evidence that when this target is either overexpressed or maybe knocked out in, let's say, naturally occurring mutants in humans, do they show an effect on a disease? Do animal models show an effect on this disease? So this really helps to, again, get confidence that your target is going to be important. Then there's help many times with how do you design your assays, and also using orthogonal assays, as in don't just measure the outcome of your drug effect on your target in one way, but do it in different ways to make sure that you're not looking at artifacts. Then many times these accelerators pull in different types of support. For example, medicinal chemistry support if you're working on small molecules, or how do you even characterize compounds? What are different aspects of a drug in development that you need to think about that maybe as an early stage discovery scientist in an academic institution, you're just not aware of? Then there's always a lot of emphasis on target engagement. So can you show in cell culture models and animal models that your drug is actually engaging with your target, binding to it, and having the desired effect? And then also, what's the selectivity there? If you're wanting to target a particular receptor and there's, for example, 4 or 5 other receptors in the same class of receptors, how do you know you're just hitting this one and not the others? Or, what other proteins in the body may you accidentally kind of manipulate as well? Is that going to cause problems? Then there's many times an emphasis, or at least an encouragement, to engage with pharma and biotech to get feedback. So if you may not be ready to outlicense a project, but having conversations, and if you get the same question back over and over again from people that are in business development and that are looking at your project, if they all asked independently the same question, it's probably something that you need to address before somebody will be ready to invest in your program. And then there's advice like regulatory scientific aspects like we just talked about. For example, we as TRxA require that you establish a target product profile. You don't need to have the answers to all the questions, but at least you need to start thinking about it. And it's an enforcing function to talk with, for example, a patient or a clinician who's treating that patient population, informing to make sure that your drug is going to solve a meaningful problem. So then, when you start from this drug discovery on the left, and then clinical trials, phases 1 through 2 and 3, and phase 4 are on the right, this middle part is what we talk about: lead optimization studies and IND enabling studies. This is where TRxA would like to come in to bridge the drug development valley of death. And our BRIDGE grants, BRIDGE stands for Bridging Research and Innovation in Drug Development Grants. We'll have to get a fun acronym, right? So TRxA funds really that piece that's really hard to get funding for. It's hard to find the expertise. It's post, I would say, typical type of NIH funds. It's preclinical funding, for example, by pharma and biotech, and it's that underfunded area where we like to make a difference. So a slide to kind of summarize this. On the left-hand side, you see a box with the problem. We just talked about the transition from academia to industry being the valley of death. A lack of understanding what data package you actually need to put together to get people interested, for example, industry partners or venture capital funds, and then the regulatory aspects of drug development as well, and then sometimes the lack of reproducibility. So then the solutions for TRxA to provide both funding and expertise, and that expertise comes in the shape of regulatory education, assisting in study design and implementation, however you need that, and then also supporting the involvement of contract research organizations to maybe repeat a key experiment in their hands, because now you have an independent third party. They're showing the same result. It's really going to convince a potential partner down the line that this is a real effect that you're looking at. And then, of course, we also actually encourage the use of CROs to execute parts of the work, if that's just done more efficiently, maybe even more cheaply at an outside institution. For example, pharmacokinetic evaluation. Unless you have, for example, a core on your campus that does PK studies all the time, you probably don't want to teach a grad student to do oral gavage in mice just for this particular PK study compared with IV. It's probably going to take quite a lot of time, a lot of animals, and it's not as efficient as sending it out to a contract research organization. Does it all the time for $1,000, $2,000 in 2 weeks. You have your answer. So this is an example where we encourage the use of CROs. So we're pretty unique in that we're an independent and not-for-profit organization. We're a neutral facilitator, and we do not claim any IP. So we're not a venture philanthropy fund. We're really a grant funder that we don't want to be a co-owner of the IP. And we will leverage the relationships and the regulatory knowledge that we have within the Critical Path Institute. And I'll talk about that in just a few slides. Then we help provide project management to facilitate streamlined planning and execution. So we meet once a month with the people that we fund. They're fun meetings. We get an update on the science, where we financially, it just helps for everybody to come together at a regular interval, have people like us who are not really on top of the project all the time, give a perspective and getting the updates about the project, and they're really engaging meetings. So how are we funded? We are funded by the Frederick Gardner Cottrell Foundation. So Research Corporation Technologies, sometimes also known as RC Tech or RCT, they're a venture group in Tucson, Arizona. And they established the Frederick Gardner Cottrell Foundation back in 1998, so it's been around for a while, and that's their nonprofit organization. So they provide financial support for scientific research and educational programs at qualified nonprofits like CPATH. So we're very grateful for the Cottrell Foundation for us to be able to start this program. So a little bit more about Critical Path Institute as a whole. So Critical Path Institute is shortened often as CPATH, and we do a lot of public-private partnerships. So this is for institution as a whole, not TRxA, but CPATH as a whole. So CPATH as a whole fosters the development of new tools to inform medical product development and regulatory decision-making. Now, how do we do that? We convene scientific consortia, public-private partnerships, with industry members, academia, government. Everybody comes together to share data and expertise. So we together build active consensus. We actively build consensus. Everybody shares in the risks and the cost of what it takes to get a tool developed. The best science comes to the table, broad experience. So when you look at the right-hand side of the slide, CPATH plays in this neutral ground where we're the convener of industry partners, regulators, patient groups, academia. And because CPATH is this neutral area, everybody comes together and is comfortable bringing their data actually to the table, to CPATH, so we can all learn from it. So point 3, you see there, we enable iterative participation of the EMA, FDA, PMDA, which is Japan's regulatory agency, to develop these new methods to assess the safety and efficacy of medical products. And then the goal is to obtain official regulatory endorsement of these novel methodologies and drug development tools. Now, a couple of examples of these tools could be a biomarker. We are working on, for example, a biomarker for how do you predict kidney injury in a human by using in vivo models, or how do you use wearable devices like your watch in a Parkinson's disease trial to look at somebody's gait? Or can we develop a questionnaire, a clinical outcome assessment for patients suffering from asthma, or maybe you have kids suffering from asthma, to track how many times they have an asthma attack? It's those kind of tools that have to get everybody together, because it's more than just one company that needs to develop such a tool. And in having everybody together, including the regulatory bodies, really, again, de-risks actually these tools, so that then drug developers can use these tools in their clinical development program. So how does this then relate to TRxA? Well, at the top you see CPath and the preclinical neutral ground with our industry partners, academic groups, regulatory partners, patient advocacy groups, and then also larger government bodies like the WHO, also NIH and CDC. They all come together and we have more than 20 consortia. So we synergize as TRxA with these consortia. Now, these consortia are focused on drug development tools, or DDTs for short, and their regulatory endorsement. And we focus directly on developing drugs. But we have all these colleagues in these consortia and connections that we can have access to subject matter expertise. And again, think about some of these regulatory aspects on the front end of starting your drug discovery program. So TRxA applies CPath's expertise to move projects to the clinic as fast as possible because patients are waiting. So then let's talk in particular about the BioBRIDGe Awards. So these are biologics-focused bridging research and innovation drug development grants. So this is new. We are announcing today a pilot protein-based therapeutics, or PBT, request for proposals. So today the RFP actually opens, and the indications are for rare and orphan diseases, neurodegenerative diseases, or pediatric indications. So we'll talk a little bit more about our typical RFP, which is a therapeutic area agnostic. This is a pilot. We need to build up our own internal skill set to support these kind of projects compared to, like, let's say, a small molecule project. And that's why we're limiting the indications here to the ones listed on your screen. The modalities that we will support in this particular RFP are protein-based therapeutics. So that means peptides or proteins, regular, bivalent, or even if you want to get fancy, trivalent antibodies or antibody-drug conjugates. And then the funding amount you can apply for this round is $250,000 US. Now, if you're somewhere else in the world, you don't use US dollars, we would just convert that. And that amount is per project for a year. And then actually, after this pilot RFP, the protein-based therapeutics will be integrated into our standard RFP that typically comes out early in the calendar year. And at that point in time, we will support all modalities, all therapeutic areas, and larger funding amounts as well. So our typical standard bridge awards are, we have 3 different types that we give out for those. So this will be, again, early next year. Our stage 1 proposal is when you're in early lead compound to late lead series stage of your project, and that's for the $250,000 that we're currently also awarding for the BioBRIDGe Awards. Stage 2 is if you're a little bit further along. If you have a late lead series and you need to select your clinical candidate, you can apply for $500,000. And then stage 3 is, okay, you have your candidate, you know what you want to take into the clinic, but you need to do some IND enabling studies, and there you can request up to $1 million. So let's talk through the eligibility criteria of the BioBRIDGe Award. It's going to be a bit of a busy slide, so I'm slowly building it up to not overwhelm you with text. First of all, the mechanism of action of the protein-based therapeutic should be known. So we're not looking for any black box of this magical protein and somehow it cures people or mice of cancer or what your favorite disease is. That's not going to fly. You really need to understand, okay, how does this work? And then we'd like to see a well-defined compound progression pathway with established success criteria in place. And such a compound progression pathway is pretty much just a series of experiments you do where you have yes, no decisions if a compound would make it through that filter does it go to the next step to be evaluated in the next type of evaluation? So typically you start with activity and then in vitro, maybe even cell-free system. Then you go through more pharmacokinetic type evaluations, and then ultimately you end up with in vivo proof of concept models. The next two on the left, you need to have profiled one or more of your protein-based therapeutics sufficiently so that the parameters that you still need to optimize can be quantitatively specified. So again, we're really looking for where are you currently at with what your profile looks like and where do you need to go, and that needs to be quantitative. And then you need to have a defined target product profile in place. This speaks to that regulatory aspect of the project that we really like to emphasize. Again, when you're early, you probably don't know exactly, how it's going to end up, but having conversations with maybe some colleagues in the School of Medicine or clinical practice to understand what is going to be your patient population and what's the current standard of care and how are you going to add to that. Having those discussions will be a very good exercise. So we ask every applicant to actually fill out such a target product profile. And we understand that this is kind of a living document. The next two are that in vitro pharmacology assays, whether they're biochemical or cell-based, for potency and selectivity, are available in either yours or the applicant's lab, or maybe a collaborator, or maybe a CRO. And they've demonstrated to be suitable to drive the characterization and optimization of the protein-based therapeutic. And then on the right, we really like to see that there's a strategy for in vivo pharmacodynamic characterization to assess target engagement and efficacy of the PBT. And if you do an in vivo experiment, we really would like you to utilize clinically relevant outcome measures as much as possible so that you have the translation to what's ultimately going to be measured in the patient, right? Could be biochemical, anatomical, or functional metrics, but showing that correlation between in vivo model and the clinical reality is going to help your project succeed. Small-scale manufacturing of these protein-based therapeutics should have been demonstrated, again, either in your lab or maybe a collaborator's lab or maybe a CRO, and then should be effective purification techniques to reduce contaminants. And then also cGMP, so GMP manufacturing should be feasible, and potential manufacturing and scale-up hurdles should be addressed in the application. So you don't need to show that you can make, 200-liter bioreactor type scale antibodies or anything like that, but you should have thought about it. What is this ultimately going to take from a manufacturing perspective? Can I make enough to do the experiments I need to do right now? And, have you maybe talked with an expert in the scale-up and purification? Because especially once you start talking about large molecules, things get kind of complicated fairly quickly. Now, on the right-hand side, we spoke earlier about the multidisciplinary nature of the Valley of Death. So the project team needs to be multidisciplinary. So it needs to include members with expertise around the target biology, the platform on which your protein-based therapeutic is based, as well as clinical practice. So you don't need to be the expert in all of that. But if you're kind of missing some parts, pull in a collaborator who at least can advise you or help you or think through some of these potential hurdles, just so that you have the complete picture from the get-go. And then lastly, a strategy is available to reduce off-target toxicity and maybe immunogenic potential while maintaining the bioactivity of the PBT. And then last but not least, the PBT is patent eligible and unlikely to be blocked by any intellectual property constraints. Again, we are funding projects where IP is already in place or can be developed. So that's something that our review group will look at it fairly carefully. So how is this going to all work? So we have a call for pre-proposals, which opens today. The deadline will be the end of June 27th, which is a Thursday. And we have a grants portal that's on the Salesforce platform. So at this pre-proposal stage, we were asking for a high-level description, non-confidential summary information about the project, like what's your target, what are the properties of your lead molecule. And this is reviewed by an internal group in CPATH called the Programmatic Review Board, PRB, and they will determine which projects advance to the next stage. So the second step is that you will be invited for a full proposal. These invitations will go out the week of August 12th. And it will be due September 26th. So you have about 6 weeks or so. So in order to make sure that we can review confidential information that's part of this full proposal, we will initiate a confidentiality agreement with your institution. And this should be in place before you submit your final proposal. And then we have an external scientific advisory committee, or SAC. They will score the proposals, and you will get the feedback from those scientific reviewers back whether you get selected for funding, yes or no. The scientific advisory committee scores and narratives will be forwarded back to this internal group called a programmatic review board, and they make the final selection for funding. This will happen middle of December. So as the TRxA team, we are not part of these PRB or SAC groups. We really want to be your champion, your mentor, your guide. So we're happy to answer any questions that you may have because we're not the reviewers. We set it up on purpose this way. But throughout the process, we welcome you to email our general email account, so TRxA@c-path.org, for any questions you might have. Due to some logistical issues, we're not offering live consultations at this point in time, although if via email we cannot sort it out, we can figure out how to jump on a call. But really, we welcome any questions that you may have, and we try to answer them as quickly as we can. So let's jump into some FAQs, because I'm sure many of you have many questions, and I'll try to answer them as we go. And of course, during the webinar, feel free to put your question into the Q&A box as well, and we'll answer them afterwards live, as many as we can. So who do we fund? Who's eligible? So trying to forward, there we go. You need to be faculty, so you cannot be a postdoc or any temporary position like that. But a faculty at a university, or maybe the equivalent of a faculty member at a nonprofit institution, but you can be anywhere around the world. And then from an institutional perspective, again, we want to emphasize that we have funding available for projects that are academic or nonprofit organizations anywhere in the world. You cannot be a member of a for-profit company, unfortunately. Because we have such close ties with CPATH, with the regulatory bodies, from a perceived conflict of interest, we cannot fund any for-profit organizations, because then people may look at us funny, like, well, if the FDA is funding CPATH and CPATH is funding a company, that seems a little fishy, right? So we're trying to stay away from it, even though the FDA actually is not funding our program. Just from a perceived conflict of interest, we want to make sure that we stay as neutral as possible. So you need to be an academic group or a nonprofit. So are there any institutional commitments required if you get funded? Well, yes. We expect to engage between institution and TRxA in a project agreement. So we have templates actually available on our website, and we expect a revenue sharing agreement. So depending on how much we've invested in a project, we ask for a percentage of any income that comes to the institution if this project gets commercialized. And we're happy to share this template. Like I said, it's on our website. We always encourage engagement with your tech transfer office, with your grants and contracts office, just to make sure that they're on board with this. Thus far, we've successfully negotiated institutions of six different agreements with 6 different institutions. So we don't foresee any problems, but still it's good for everybody to know up front what the strings are that are attached to this grant. So again, we don't claim ownership of any IP, but we do want to have that revenue sharing agreement in place. So what do we fund then? So what therapeutic areas, again, are eligible for TRxA BioBRIDGe awards? Because I'm sure that flashed by on your screen fairly quickly. So we are looking at this round for the following indications. It needs to be a rare and orphan disease, or a neurodegenerative disease, or a pediatric indication. Now you can even have projects that fall into one or in 2 or more of these actually, but it needs to hit at least one of these different therapeutic areas. And what are the criteria then? What constitutes, in TRxA speak, a protein-based therapeutic? So our definition right now, and I'm sure there will be gray areas where people are like, well, how about this? But it needs to be either a peptide or a protein. Amongst the proteins is, regular, bivalent, or trivalent antibodies, and also antibody drug conjugates are eligible. So if you're like, well, how about this particular flavor? Reach out to us on our email address, and we will try to address that question that way. But hopefully this will give you enough guidance to know whether you're in or out. So what level of funding is available for these TRxA BioBRIDGe awards? Because this is a pilot, we're only offering the $250,000 grant award, which is still, a meaningful chunk of change to move the needle on a project, we hope. We do limit indirect costs to 10% and pass through costs to service providers like a CRO or a consultant. They're not eligible for indirect costs. And then some people ask, well, does the TRxA funding all come to my lab, or how does this work? And we want to emphasize that the funding is for your project, not necessarily your lab. So it could be, of course, you're used to moving money to collaborators' labs, but also if you use a CRO or consultants, that's all part of your whole $250,000. So we don't really care. It sounds so negative, but to us, it's immaterial where the work is done, as long as the work gets done. So whether that's in your lab or collaborators or the CRO, it's all okay with us, and we're happy to support that. But again, making sure that you're aware that the 10% indirect cost is not allowed for those pass-through cost to service providers. Speaking of allowable costs, so we are happy to support costs associated with defined project tasks, like, of course, lab supplies, animal procurement, core services that you may use. What is not allowed are general lab expenses. So if you need to buy new lab coats for your whole lab, you need to use a different fund for that. Travel is not covered either. Neither is equipment, tuition, or IP prosecution and maintenance. We get this latter question quite a lot, actually. Well, can you help us fund, our IP? But because we are not taking any ownership in that, so this is, we leave that squarely with your institution, your tech transfer office. And then we've had the question, well, do I need to worry about having other funding sources for my project? And in fact, no, that's a good thing. We actually like to partner with other funders. Of course, we don't want you to double dip and get paid twice for the same work, but there's always more work that needs to be done in drug discovery projects. So if you have already a federal grant, or maybe a nonprofit is funding part of your research, and you need us to come in to do another piece, or maybe once we come in, of another nonprofit or other group that may want to fund, we're happy to have a conversation and see how can we maximize the impact of our respective funds. And if we all pool it, we will probably get a better return on investment, both from a results perspective, but also maybe from a financial perspective together. So we're happy to partner with anybody that you think might be also interested in funding your program. Then a few questions about CROs and intellectual property. So if some people wonder, well, how do I find the right CRO if I even need one? Like, I've never worked with one. Well, we're happy to point you in the right direction. So if you require advice, TRxA can help identify suitable CROs to do part of the work. We have no geographic restrictions. Management of the contract will be through your institution, though, but we're happy to help manage the work to be done. We've had some examples where Mark has provided some suggestions what chemical compounds could be made to complement what the PI was thinking of. And then we helped get some quotes from a couple of different vendors and see which one made the most sense for that particular project. So we're happy to be an active partner in that process. And then we also get some questions around if you need quotes, because I think for SBIR, STTR funding from the NIH at least, you do need quotes. We actually do not require them. So we do actually have some ballpark numbers available for you for standard drug discovery type assays like a selectivity panel, PK studies. You don't need to put a budget together until the full proposal stage. And at that point in time, we will provide this kind of menu to you that you can use to build your proposal. Of course, if you have quotes because you're already working with the CRO, totally fine to use those numbers as well. But if you don't, we don't want you to use your time to reach out to a lot of vendors and get a sense of that, because we have that information that we can share with you. And they're ballpark numbers. We're not trying to breach confidentiality with the exact numbers that we get, but we've rounded them up some and made a bit of an average. And then the question is, do I need to have IP filed on my compound before applying? Actually, no. So there's no requirement that IP protection is in place, but rather that the composition of matter is eligible for protection. Now, it's okay if you have IP already either filed or even awarded. Although if it's kind of old, then you may want to think about, okay, is there new IP that you can file, because it takes so long to get to the other end, right, of getting an approved drug, that you want to make sure there's enough patent life left at that point in the game for groups like TRxA and follow-on funders to be excited about it. But we do expect IP to be filed internationally, not just in the US, and this is mostly relevant for US-based groups. Some universities and tech transfer offices, to save money, sometimes have the habit of only filing in the US jurisdiction. That becomes a problem later on, because if you don't have international coverage, a potential licensing partner or a venture capital group is not going to be interested in that asset. So that's why to help you protect yourself, really, we require this more international filing of your IP. So how do I apply, right? So you told us about this awesome grant. So the question is now, what do you want to do next? So first step is to create an account in TRxA's grant portal in Salesforce. So the link can be found at the TRxA website. The TRxA website is just easier to remember than the grant portal website. So we're at c-path.org/programs/TRxA. And then on that website, there's funding opportunity information. If you scroll down, there will be a link to the grant portal. So you need to set up a username and password. The portal will automatically close at midnight in your local time zone on the day of the deadline. So please start early. There's always little snafus that come up with these kind of electronic portals. So I really encourage you to A, set up your user account as early as possible, and B, get a little bit ahead of the game and maybe not wait till 1 minute before midnight to submit your proposal, because you may run into an issue, and then, you know, we may not be in a time zone compatible with yours that we're available to help you work through it. And also, for some technical issues, we are relying on the Salesforce team. So, you know, so start early and work through it before you get to crunch time. And how many projects do we fund? So in this particular cycle, we anticipate funding 2 to 3 projects in a special funding cycle. In our typical cycle, we fund between 4 and 7. This is a smaller, again, a pilot cycle, so that's why it's a fewer projects. And then some people are wondering, well, what's my chance of success? Well, this is our first time, so we don't know. As a data point, again, how relevant it is, we'll see. For our regular RFP earlier this year, that was for small molecules, we received 80 pre-proposals, but these projects could cover any therapeutic indication, and again, it could only be a small molecule. So this is slightly different because we have narrowed the scope regarding therapeutic area, but broadened the platform. So we will be waiting along with you how many proposals we get. So then I want to end here with the call to action. So the next step, again, is to create your account in Salesforce. Again, here's the website, c-path.org/programs/TRxA. Submit your proposal by the deadline. And then if you have any questions, email us at trexa@c-path.org. We're happy. We try to answer within a business day or so. We're usually pretty quick. Again, we have a couple of travel issues coming up, so that's precluding us from doing a lot of live consultation, but we'll try to get everything clarified via email, and if we really need to get on a call, we'll make that happen. So then I want to leave you with this resources slide. Again, the website address is here on the top. On the website, you can also find a guidance document, and this guidance document lays out what are the scientific review criteria, what are the deadlines again for everything, how does this work, how does that work. It's a very comprehensive document with a lot of information, so I really encourage you to read it. There probably any question you have, 80% chance the answer is going to be in that guidance document. There's also a user guide that's a separate document for Salesforce, so for the grants portal. So it will have step-by-step instructions how to create an account, how to submit your proposal, and so on. And then we also have this agreement template, and this will be very interesting to your tech transfer office, to your grants and contracts office, because they will want to see what are the strings that are attached to that. Our email, again, is trexa@c-path.org, or you can also email me personally at meverts@c-path.org. And then also put my LinkedIn profile there. I also want to point out on the right-hand side, you'll see I'll be at Bio with several of our leadership. So if you are heading to Bio, reach out through the partnering platform, and I'm happy to connect there. And then finally, at the bottom, there's an article that Mark and I published last year. It's open access, so it's free for you to download. Successfully Navigating the Valley of Death, the Importance of Accelerators to Support Academic Drug Discovery and Development. So there's a more comprehensive explanation of what different accelerators can do to de-risk a project. So I'll leave this up for a few minutes. At the same time, I'll ask my colleagues to come back on video, and we can deal with a couple of questions as they came in.
We have some questions coming in. So I'm going to, these first couple of questions, I'm going to ask you, Mark, if you can, respond on behalf of the team. How far into the lead selection process does the protein-based therapeutic have to be for the early lead optimization stage? Are molecules that have shown to be target-specific eligible, even though they have not yet gone through a full functional assay evaluation?
This is for me.
Yes.
Oh, thank you very much. It's a good question. So I would say I'll let Micah correct me if I'm wrong, but they would be eligible. I think you'd have to provide for the reviewers some evidence why affecting this target is going to be affect the disease. So if you've got target engagement, that's great. But then genetic evidence of why this is a target of interest would be supportive of the application.
Yeah, and I would add knowing what potential issues are with your protein-based therapeutic and having a plan to address it. So no molecule is perfect from the start. So it may be that you need to have codon optimization, or maybe charge islands need to be addressed. Having an awareness of it, it doesn't need to be fully characterized, but at least showing that you've thought about this and that you plan to address it in this proposal, or maybe not yet because it's something for later, but it shows that you've thought through this whole process, and that will really play well during the review process.
Just want to be sure that everybody sees the answer to. There was a question about whether nanobodies are eligible for BioBRIDGe awards, and the answer to that is yes, they would be eligible. Okay, another question we have, are murine monoclonal antibodies that have not yet been humanized eligible?
I'd say yes. If you, again, because many times you need kind of one of each, right? Because you need to do your, especially if the target is not a homology and you need to have a separate antibody for your mouse model or your animal model in general and a human one, that would be eligible, because sometimes that humanization part really, it's not going to do you any good to get proof of concept in an animal model. So yes.
We also had a clarifying question about whether gene therapies expressing a therapeutic protein are acceptable for this pilot mechanism. They are not in scope for this pilot, but they will be in scope for our next RFP, our standard RFP that will be announced in January 2025. Okay, I have a question here. Maybe Micah, you can take this one. Does this grant opportunity include interest in emerging virus antibody therapeutics?
Well, I guess it depends if your virus infection does something to neurodegeneration or is a problem in pediatric populations. So I think you need to make the case why this is important for those therapeutic areas. So anti-infectives, it's not as a blanket encompassed in this particular RFP. But again, if you're causing a neurodegenerative problem, then I could see, although maybe it's going to be a bit of a stretch, I think your best bet is if let's say you're working on RSV is a huge problem in pediatric population, you would be eligible, right? Because we're covering neurodegeneration, rare and orphan diseases, and pediatrics.
Thank you, Micah. We have a question here. How can we check whether our target disease is considered rare or orphan disease? And I think our answer to that would be the definition, the is that the CDC's or NIH's definition of a rare and orphan disease, which I believe is fewer than 250,000. But if in doubt, please email us at TRxA@c-path.org, and we can help answer that question for you.
Yeah, and I know the EMA has a slightly different definition than the FDA. So if it's on the border, we'll probably call it rare, right? But look at those definitions of your potentially maybe your local geographic area. And if you're just outside, maybe look at the other one and see if then it falls in or not.
Great, thank you. But we have a question here that I don't think we touched upon. There's interest in knowing what the reviewers' criteria will be when they're looking at the pre-proposal applications. Mark, do you want to guide us through that?
Yeah, I can give it a shot. Yeah, so there's about half a dozen criteria. We're looking for a project that meets an unmet medical need. The world doesn't need another statin, so you know, a disease that's really with a need for the patient. Novelty. We're looking for a project that's not right in the middle of what big pharma's doing. We don't want to be competing with pharma. We look at commercial viability because we want to get to the next inflection point where a fund is going to come in and be interested in it. So there's got to be a path forward to market. We don't want to be in such an ultra rare disease probably where when it's going to be an issue to advance the project. Obviously, sound scientific rationale is a big one. If you can concisely communicate with the reason for doing this project, that's going to resound with the reviewers. And obviously things like, you know, a well-structured project plan, you know, showing that you know what you need to do to move forward. And then there's things like, you know, we look at the likelihood of success. You know, how difficult is this going to be to advance to the clinic? And you know, is the team in place? Is the funding appropriate to get you to that inflection point? And that's kind of the main things you need to hit in your application.
And if I may add to that, many people, at least in the US, are used to the NIH proposal structure, which is typically 3 specific aims, and they need to be independent from each other. Otherwise, you get creamed by the reviewers, right? We're kind of the opposite. So we actually structured both the pre-proposal and the full proposal as a series of questions we'd like you to answer with a certain character limit that you can paste into the Salesforce platform. Drug discovery is not, we actually want to get away from those 3 independent specific aims because everything depends on each other in drug discovery. We know that. So once we award a project, we actually together come up with the payment plan that's milestone based, because we know things change as you kind of go through the process. So it's perfectly okay to have a very linear, actually compound progression pathway where I'm doing A, B, C, and then D. And you know, through it all, you'll probably have multiple candidates and some will fall out because they don't meet certain criteria. So that is actually the more organized and stepwise you are in your proposal, that tends to speak very well to our reviewers. So try to get away from your NIH mindset a little bit. And again, we try to guide you towards what we like to see with the questions that we ask. We still often see people reverting back to that 3 specific aims because it's just been so hammered into them. You don't need to have 3 independent specific aims. It can be very linear.
Question, I think. Here comes another one. Micah, maybe you can take this one for us. Do antibody drug conjugates include antibodies conjugated to a peptide, not a small molecule?
Yeah, I think so. Sorry,
Okay.
I forgot. I didn't hear who you were referring, but yeah, that would be eligible. That sounds intriguing. Yeah.
Yeah. Excellent. And Elizabeth, thank you for your kind words. Elizabeth asked if we can share the slide deck recording with participants, and we most certainly can do that.
Mm-hmm. Yeah, and we also recorded this, right?
Yes. It's been recorded and it will be posted to our website, and you'll also receive a notification that the recording is available, and please feel free to share it with any colleagues who you think may be interested. Okay, so that wraps up our questions. So I think we can give everybody a few minutes back to their day. Thank you to everyone on the TRxA team for the great overview and the responsiveness to questions. And thank you so much to everybody who joined us today. We appreciate your interest, and we look forward to seeing your pre-proposals in the portal.
Yep, we'll learn together. Have a good day. Bye.
Bye-bye.
Thank you.